So this is another animal cell model, and we'll move a little bit closer to look at the organelles. But let me show you a few things while we're here. This structure out here is all cell membrane, that phospholipid bilayer that helps to control what goes into and out of the cell. It would have proteins embedded in it that also serve as part of the glycocalyx that helps to identify this cell to other cells, or those proteins can serve as enzymes in the cell membrane. They can serve as channel proteins or transport proteins. They can help adhere this cell to other cells. This structure is the nucleus. And these pores that you see, these dots, those are the nuclear pores. So it wouldn't really be open in the front here. Those nuclear pores sort of act like doorways that allow synthesized rRNA to leave the nucleus, or it allows maybe nucleotides to get into the nucleus so that you can do DNA synthesis or you can make ribosomes. This, what looks kind of like threads here, is chromatin. So let's go in and take a closer look. The chromatin is your DNA. So it's chromosomes, but they're in a thin form. They're not in that tightly coiled form that you'd see in late prophase or metaphase, anaphase, telophase of the cell cycle, because this cell would be resting at the moment. Here's 1, 2 nucleoli. Singular is a nucleolus, and their job is to manufacture rRNA, ribosomal RNA, that becomes part of the ribosomes. And there's some here, there's some up here, and you can also see them over here attached to rough ER or rough endoplasmic reticulum. The job of the ribosome is to synthesize proteins. The rough endoplasmic reticulum here has an important role in synthesizing and modifying proteins that might be used in this cell, but generally they're often destined to leave this cell and go out to other parts of the body. So if this was, say, a liver cell, that rough ER might be making albumin, which helps us to maintain water balance between body cells and the blood plasma. These orange structures here, there's a whole bunch of them. Those are mitochondria. Mitochondrion is singular. The mitochondria are the sites of aerobic cell respiration. It's where we can extract the most energy from, say, fatty acids or glucose, because we're using oxygen to make that ATP. This one and this one are peroxisomes and lysosomes. And it's a little hard to tell just by looking at them which is a peroxisome versus which is a lysosome, but they have pretty similar functions. They both contain hydrolytic enzymes, enzymes that will use water to break down chemicals, break up, say, pathogens maybe that were endocytized by this cell, or to break up toxins or chemicals. Lysosomes and peroxisomes can work with the smooth endoplasmic reticulum. And smooth ER has a lot of different jobs, but let's just talk about a few. So one of those jobs is that it can help detoxify substances that have come into this cell. So that might be the byproducts of a medication, those, say, inactive ingredients. It might be a poison that's come into the body, or a poison that's been produced by an antigen or a foreign invader to this body. Smooth ER is also incredibly important in synthesizing lipids for this cell, including phospholipids that become part of the cell membrane. Another thing that smooth ER does, and in particular there is sarcoplasmic reticulum in muscle cells. So in muscle cells, smooth ER takes on a specialized form where it stores calcium ions, and those calcium ions are critical to muscle contraction. Over here, this is another structure of the endomembrane system. This is the Golgi body. You can also call it a Golgi apparatus or a Golgi complex. It just depends on the text that you're looking at and what you see coming into it and coming out of it. And if you follow, you can see that this vesicle is going to here fuse with the cell membrane and allow exocytosis of a product that was modified and packaged in that Golgi body. Some people think of the Golgi body as sort of like a post office where there's mail that comes in and then the Golgi helps to figure out where does that mail need to go, or maybe you have to package up that mail, especially that would be something the Golgi does as well, and then sets it off for export, whatever that chemical or product might be. Over here, these two are the centrioles, and one of their big jobs is to make spindle fibers. Those spindle fibers will attach to the sister chromatids during mitotic division and then help, sort of like fishing line, pull them, the chromatids, to opposite sides of the cell so that they can become part of two new cells, and we get an equal number in the same chromosomes in each different new cell.